Okay, we're live. So, greetings, everyone. I apologize, my camera does not seem to be on, but I think you can all hear me. I'm Peter Young. I'm a board member of Societe de Industrial, and I'm head of programs. And I'm very pleased to be the introducer for today's a webcast uh, on recombinant DNA and the birth of biotech. And David Cole, who is the CEO of the Science History Institute that I once was on the board of, and is going to present. And then we're going to have some discussion uh, right after his presentation, and then we're going to have Q&A for all of those of you who have questions that you want to ask. Two things logistically, and that is that if you have any questions that you would like to present to our speaker, you can do so just by using the chat function. And both David and I will be able to see your questions, and we'll field whatever questions that we can. Also, a unique feature of this particular technology platform is that at the end of David's session, we're going to put all of you into a essentially a virtual cocktail space. So what you'll do is when we end, and there'll probably be some link you might have to click on called Fluid Space. You may not. Once that you do that, though, you're going to be essentially in a virtual networking arena, and you will be able to move around in the space with your arrow keys, and you can approach people and talk to them. And as you get closer to them, you'll be able to hear them. As you move away, their voices will be more distant, as is true in real life. The other thing I should mention is that if you want to know who each person is, there will be a name associated with their picture, but you can also hover your mouse over their picture, and it'll tell you the name of the person, their title, and their company if you want to be able to just see who they are. Hopefully, you'll have some fun. David will be in that fluid space, so you can talk to him afterwards. But the main thing is it's going to give you the opportunity to talk to the other people who are attending this event, and that will last at most half an hour, but just give you an opportunity to talk to the speaker, but also to talk to other people who are in attendance. The last thing I want to say is that we have a number of wonderful events coming up. We have one in April on women in chemicals in the chemical industry. So we'll have a panel, but also like this, there'll be opportunity for the people who register to talk to each other, network. We're very excited about that. And there'll also be one on May 1 on developments in construction chemicals. And one of our board members, Sean Pan, will be leading that panel. And that's an area where there's a lot of innovation and a lot going on. So I encourage you, if you're interested in that area, to attend. But obviously, you'll get meeting notices for those events. David Cole, I've known for a number of years. He joined as the CEO of the Science History Institute, I guess about 3 or 4 years ago. Is that right,
Yep,
David?
yep, that's right.
And he has really done wonderful things on what has always been a very successful organization. But he's done a lot of things to innovate and to essentially connect the Science History Institute to a broader audience. I'll let him talk a little bit about that. But of course, to have him as a speaker on this topic is perfect, because not only is he going to talk about the history of DNA, which is a really critical innovation for a whole lot of industries, and particularly the biotech and drug industry, but also it continues to evolve, and so there are a lot of things that are happening around DNA, recombinant DNA, that are going to drive a lot of innovation going forward. So he's going to talk both about the history, which is fascinating, but he's also going to talk about the future, which is even more fascinating. So with that, let me turn it over to, oh, and I do have one last thing is that we have our International Palladium Medal Dinner Award, which will be on May 8th. And we all encourage you to attend. We're going to be honoring Jim Fettering, who's the CEO of Dow, and we'll have a, it'll be at the New York Hilton, and it's going to be a wonderful event and a great opportunity to both hear a couple of speeches, but also to meet a lot of people who are in the industry or serve the industry who will be in attendance. And you can register for that on our website. So with that, let me turn it over to David, and he has some slides, but also we'll have some discussion when he's gone through his slides. Go ahead, David.
Great, and just thank you. And Peter, I just want to confirm that you can see my slide deck right now. Is that correct?
Yes.
Okay, very good. I hope everybody can hear me. Well, good afternoon. As Peter mentioned, I'm David Cole, and I am the CEO of the Science History Institute, as Peter mentioned. And in fact, it's through that connection that, as Peter mentioned, that he and I have known each other. And we like sharing ideas back and forth, what's up at our respective institutions. And he was gracious to invite me to be with you today to share a little bit about the history that we cook up at the Science History Institute. At the Science History Institute, we tell the stories behind important developments in the history of the chemical and the life sciences. As historians, we want to use the past to illuminate the origins of and to make better sense of the science and technology that has our attention in the present. Perhaps no scientific development that compels our attention today is more worthy of historicizing than the astonishing work being done in gene editing. In a recent article, the New Yorker magazine described CRISPR-based gene editing techniques, most closely associated with the work of scientists Jennifer Doudna and Emmanuelle Charpentier, pictured here, as, quote, perhaps the single most transformative biological technology of the 21st century, end quote. Doudna and Charpentier, winners of the Nobel Prize in Chemistry in 2020 for their work on CRISPR-based gene editing, have been rightly lauded. Along with Feng Zhang of the Broad Institute, they've been lauded for paving the way for the development of precision gene editing techniques that are driving the promise and increasingly the reality of transformative interventions in areas such as biomedicine and agriculture. Now, CRISPR has been very much in the news since Doudna and Charpentier made headlines for their pioneering research on its potential as a gene editing tool. In 2012, they and their teams developed a method of repurposing a bacterial immune system called CRISPR. This is an acronym, CRISPR, that stands for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPRs are repeating segments of genetic code on DNA, interrupted by so-called spacer sequences. These genetic passages, if you would, serve as a genetic memory that helps bacterial cells, for example, detect and destroy harmful viral invaders. A CRISPR-based system can make breaks, cleavages in DNA at very precise locations using a CRISPR-associated enzyme, such as the Cas9 protein, like a pair of molecular scissors to cut DNA. Scientists can now edit the genomes of living organisms by adding new fragments of DNA for cells to use as templates when they repair breaks in their DNA. In this way, scientists can, for example, replace a disease-causing mutation with a healthy genetic sequence, or they can make other modifications to the genome. Scientists can also use this CRISPR method to knock out a gene entirely, a technique that is frequently used to study the functions of genes, or to modify portions of the genome that affect how genes are expressed, known as epigenetic editing. Together, these methods are giving scientists powerful new tools to treat disease, improve agriculture, and study fundamental questions of biology. The sheer number of potential applications of CRISPR-derived gene editing is astonishing, and they may be the single largest drivers of life sciences innovation and commercialization for years to come. In just one recent example of this potential, Jennifer Doudna's Innovative Genomics Institute has created a CRISPR-based therapy for sickle cell anemia, a genetic disease that affects millions globally and is caused by a single-letter DNA mutation that causes blood cells to become distorted and to block blood vessels. Doudna's team has developed an experimental therapy that involves 1, collecting and isolating a patient's blood stem cells, 2, using a brief electrical current to introduce CRISPR enzymes to the extracted stem cells, along with a template for correcting the sickle mutation, 3, using chemotherapy to make space in bone marrow for the edited stem cells, and 4, reintroducing the patient's own edited cells. On December 8, 2023, the U.S. Food and Drug Administration approved Casgevy, this groundbreaking CRISPR-based gene editing therapy from Vertex Pharmaceuticals and CRISPR Therapeutics. It was approved for sickle cell disease. The approval was never seriously in doubt, as the therapy demonstrated spectacular clinical results dating back to the first patient, Victoria Gray, who was dosed in July 2019. There is a certain irony that the first approved CRISPR therapy from a technology barely a decade old should be for the genetic disorder that Linus Pauling famously dubbed, quote, the first molecular disease almost 75 years ago. But while scientists have only begun to tap the potential of CRISPR-based gene editing, Jennifer Doudna will be the first to acknowledge that the process of researching and developing this technology for positive applications has been fraught on many fronts. The CRISPR phenomenon has bred intense scientific competition among CRISPR-focused laboratories around the world, and it has fueled fevered races for intellectual property rights to and commercial applications of these technologies. It is also at the root of national government-directed efforts across the globe to achieve supremacy in life sciences innovation. And this race to unlock and apply the potential of gene editing raises concerning and even frightening ethical questions for researchers, governments, companies, and members of a hopeful but wary general public. In fact, in interviews with her biographer, Walter Isaacson, Jennifer Doudna recounted a nightmare she had over a decade ago. In this nightmare, Adolf Hitler appeared to her and wanted to learn more about CRISPR and how to use it for the Nazis' terrifying eugenics program. Inspired by the potential prospect of unsafe, immoral, and even horrifying applications of gene editing technology, Doudna resolved to lend her perspective and voice to a global effort to consider the ethical ramifications of gene editing. In collaboration with a stellar team of international scientists, government officials, entrepreneurs, and journalists, Doudna helped organize the first international summit on human gene editing in 2015. This event was followed by another, just like it, in Hong Kong in 2018, at which the fears of many scientists and policymakers worldwide were realized. The 2018 Hong Kong summit was thrown into an uproar by a presentation from a young Chinese scientist, He Zhongqi, known to his colleagues as JK. A brilliant one-time Stanford postdoctoral fellow in bioengineering, JK was directing a research program at China's Southern University of Science and Technology in Shenzhen, China, where he was tackling an incredibly ambitious project, using gene editing to help parents with the AIDS virus give birth to AIDS-free children. HIV enters human cells via a receptor created by a gene called CCR5. Scientist JK used CRISPR technology, or what he liked to call God's magic scissors, to disrupt gene CCRD in human embryos, rendering babies born of at least one parent with AIDS immune to infection, and protecting their future offspring from the virus as well. Just days before the Hong Kong summit, JK had released videos announcing the birth of a set of twins in China, whose embryos he had edited with CRISPR technology. At the summit, JK said that the children were healthy and that his research marked the dawn of a new age of embryonic, heritable gene editing that promised to end many diseases plaguing humankind. The scientists in attendance were horrified. In 2018, as is the case today, the strong consensus in the scientific community was that using CRISPR gene editing to treat existing individual subjects with compelling medical conditions was permissible. But editing human embryos for disease-free reproduction is taboo. The possibility is simply too great that a scientist will accidentally introduce mutations that harm the subject and then go on to be inherited by future generations. Scientists believe that JK had misused a powerful technology and gambled with the health of children, creating experimental subjects himself without any urgent medical reason to compensate for the unknown risks of editing mistakes into the human germline. Jennifer Doudna and other leading genetic engineering experts had foreseen this disturbing development. In a 2015 article published in Nature, a group of scientists wrote an article entitled, "Don't Edit the Human Germline," in which they argued that the controversy over editing human embryos would imperil the prospects of editing in so-called somatic or non-reproductive cells, which could save the lives of millions of people who were already alive and suffering. In addition, scientists were concerned that CRISPR technologies do not always behave predictably. They sometimes cut targeted genes and then keep on cutting, leading to so-called off-target mutations. And occasionally, disabling a gene can solve one health problem while creating another unanticipated one. For example, the gene that protects certain populations in the global south against malaria can also code for the development of sickle cell disease. Scientists are loath to unintentionally engineer such possibilities in the genome. Jennifer Doudna, Emmanuel Charpentier, and their teams shared these concerns early on in their CRISPR research. In 2014, Doudna read a paper by a group in China that had used CRISPR to modify monkey embryos and then implanted them in a mother, creating a mutant offspring with altered DNA. Doudna felt that this was an obvious indication that human germline editing was coming next. In 2015, Doudna convened a group of scientists in Napa Valley to discuss the ethical challenges posed by gene editing. It was this meeting that led to the first international summit on human gene editing in Washington later that year. Significantly, two of the scientists invited to this smaller Napa convening were Nobel Prize winners, David Baltimore on the left and Paul Berg on the right. At the time of this convening, David Baltimore was 77 and long retired from the presidency of Caltech. Paul Berg was 89 and 15 years retired from his academic posts at Stanford. But Baltimore and Berg, along with their legendary expertise in genetics and genomics matters, had something else important in common. Both had been principal conveners of a now legendary scientific conference held in 1975 at the scenic and rustic Asilomar Conference Center on California's Monterey Peninsula, which you see here. Asilomar Conference 1975 was an unprecedented gathering of not just prominent gene scientists from all over the world, but also, and crucially, lawyers, government officials, and, as we shall see, members of the press. Today, we are almost 50 years removed from the 1975 Asilomar Conference. One may ask why a scientific conference held in 1975 is worth our attention today, as we consider the present and the future of the gene editing revolution. But as an historian, curious about science's past and how it might be shaping scientific developments today and tomorrow, I subscribe to the view that the 1975 Asilomar Conference was a watershed, a transformative moment in the history of science in the 20th century, a moment which shifted the course of debate about the relationship between science and society, and whose aftershocks are still being felt today in the world of gene editing and beyond. To understand why Asilomar 1975 was so pivotal and worthy of historical attention, we need to review the stunning breakthroughs in two related technologies that emerged in the early 1970s, recombinant DNA and DNA cloning. The story of recombinant DNA is very much a Bay Area story, and even more particularly, a Stanford story. In 1959, Paul Berg, pictured here, joined the faculty of Stanford University, where he became interested in the genetics of microbes and in learning the techniques of animal cell culture. Like other scientists in the 1960s, Berg wondered if it could be possible to insert foreign genes into a virus and then to use this virus as a vector, a vehicle, if you would, through which DNA and genes could be carried into new cells for deeper investigation. In 1971 and 1972, Berg undertook a landmark gene splicing experiment, which involves splicing a segment of the DNA of a bacterial virus, a virus known as lambda, into the DNA of a simian virus called SV40, whose natural host is a monkey. The DNA of both of these viruses is contained in closed loops. In the first step of Berg's experiment, the loops were each cut in one place by an enzyme called EcoRI, which was isolated for this purpose. Next, to make the ends of these now linear DNA molecules stick together, they were modified by two other enzymes using a procedure developed by Berg's Stanford colleagues. Then, the two different strands of DNA were mixed together, where they rejoined into loops in such a way that the new loops combined DNA from each source. Berg's gene splicing experiment resulted in the first human-made recombinant DNA, or rDNA, as such molecules came to be called. For this achievement, Berg would share the 1980 Nobel Prize in Chemistry, along with Walter Gilbert and Frederick Sanger. But Berg's initial reaction to his rDNA accomplishment was cautious. In spite of the entreaties of groundbreaking students in his lab, such as Janet Mertz, who was carrying complementary research forward at a rapid pace, Berg did not immediately take the step of introducing his newly made rDNA into another organism. He expressed concerns that unfettered pursuit of this research might create unforeseen and damaging consequences for human health and for the Earth's ecosystems. One fear was that rDNA carrying a dreaded gene, for example, a gene for the creation of cancerous tumors, might escape the laboratory in some common bacteria and be spread elsewhere. Berg's concerns were further amplified when two other researchers, Stanford's Stanley Cohen, pictured here with his brilliant research associate Annie Chang, and UC San Francisco's Herb Boyer, made another stunning research breakthrough, a landmark in the development of modern biotechnology. the insertion of recombinant DNA into bacterial cells in such a way that the foreign rDNA could replicate naturally in the bacterial cells. How did this happen? In November of 1972, Boyer and Cohen were attending a conference in Hawaii on the biology of plasmids. Plasmids are small, circular pieces of DNA that exist separately from chromosomal DNA, typically in prokaryotic organisms such as bacteria. Plasmids often carry genes that confer special survival advantages for bacteria, such as antibiotic resistance, and they can replicate independently of a bacterium's chromosomal DNA. Over sandwiches late one night at the conference they were attending, Boyer and Cohen laid plans for a collaborative project to discover what genes are present on plasmids and how they are arranged. The ensuing collaboration between Boyer and Cohen was very close. Plasmids isolated at Cohen's lab at Stanford were transported to Boyer's lab in San Francisco for cutting by the EcoRI enzyme and for analysis of the DNA fragments. These were transported back to Stanford, where they were joined and introduced into bacteria, E. coli, where they multiplied or were cloned in their new bacterial host. In 1973, Cohen and Boyer next showed that using their rDNA cloning techniques, the DNA from an animal, a South African clawed frog, could be inserted into and propagated in E. coli. What Cohen and Boyer had demonstrated was that transgenic bacteria, cultivated in large numbers, could effectively function as replication factories for desirable genetic material. Commercial ventures quickly started up with the objective of capitalizing on Boyer and Cohen's new DNA cloning technology, despite growing concerns over the technology on the part of scientists and public officials. At the forefront of these ventures was Genentech, the first biotechnology company founded in 1976 by Boyer and a young venture capitalist, Robert Swanson, to find medical applications for Boyer's and Cohen's DNA cloning technology. And it did not take long to find such an application. In 1978, Boyer collaborated with two scientists at the City of Hope Medical Center in Duarte, California, Keiichi Itakura and Arthur Riggs. Together, Itakura and Riggs, with Boyer, constructed a plasmid that coded for human insulin, which could then be replicated in bacteria and developed into synthetic insulin for the treatment of diabetes. Genentech then signed a licensing agreement with Eli Lilly to develop the production process for this synthetic insulin. Following this 1978 discovery, in 1982, Humulin was approved by the FDA, and it became the first biotechnology product to appear on the market. But as I noted earlier, 3 years before rDNA and DNA cloning enabled the launch of Genentech and indeed the biotech revolution, Paul Berg had qualms about the deployment of these promising but worrying new technologies. In June 1973, Stan Cohen and Herb Boyer announced that their DNA cloning technique at a Gordon Research Conference, June of '73. The possible biohazards of this new technology alarmed many of the scientist attendees, prompting two of them to send a letter to the American National Academy of Sciences a few months later, sharing their concerns and recommending the creation of an advisory committee to examine the potential dangers of creating new genetic material. In response to this letter, the National Academy of Sciences asked Paul Berg to lead a panel on the new technology. This panel, which included prominent molecular biologists and bioethicists, met in 1974 to discuss the implications of rDNA technology and DNA cloning, and to discuss the possible regulations which the government could theoretically impose on experiments using these technologies to guard against biohazards. After lengthy deliberation, one of the participants declared, quote, "Well, if we had any guts at all, we'd just tell people not to do these experiments. Maybe what we ought to do is make some public announcement," end quote. Although the panelists were uncomfortable with the principle of limiting scientific research, they ultimately agreed to publish a letter asking for a temporary moratorium on rDNA research. The Berg letter, which I show you here, as it came to be known, is viewed by historians as a rare instance of an expression of alarm about research from scientist to scientist and from scientists to governing bodies. Among the few analogous examples of such action, we might include Isaac Newton's 1676 letter to the Royal Society and to Robert Boyle, in which he recommended the cessation of open communications of Boyle's alchemical research. In the 20th century, the letter drafted by Leo Szilard to President Roosevelt on behalf of Albert Einstein, communicating deep concern about the possibility of the development of an atomic bomb, would be another rare example of this type of communication. The Berg letter was recognized instantly as a unique happening and received immediate attention around the globe. An advisory committee was formed on rDNA technology in the UK, and reactions to the letter were published in France's Le Monde newspaper for the public to see. Most importantly, the Berg letter announced the organization of an international scientific conference the following year for the purposes of evaluating and discussing the potential biohazards of and possible regulations on rDNA and DNA cloning. Amid growing scientific and public fears of superpathogens or laboratory leaks, Paul Berg, David Baltimore, scientists Maxine Sanger, Sidney Brenner, and other prominent scientific colleagues gathered in February 1975 at the conference center at Asilomar State Beach near Monterey. They gathered to debate the big questions and discuss next steps. Asilomar was an auspicious choice for this gathering, a remote location away from campuses and government agencies where the scientists could engage privately in wide-ranging and often heated debates about the implications of the new technologies. Crucially, the gathering and discussions included not only prominent life scientists, but also invited lawyers, political figures, including members of Senator Edward Kennedy's staff, government officials, and members of the press. In fact, journalists comprised 10% of the attendees. One science journalist told the Asilomar organizing committee, quote, "A secret international meeting of molecular biologists to discuss biohazards? If the press isn't allowed, I'll guarantee you nightmare stories," end quote. A number of prominent scientists, including James Watson, co-discoverer of the structure of DNA, were outspoken in their opposition to any restrictions on rDNA research, scientifically or government mandated. Tensions at the conference ran high as different speakers clashed over issues about the freedom of scientific inquiry and the social responsibility of scientists to safeguard the public from dangerous inventions. rDNA technology was so, quote, ridiculously simple, end quote, as Paul Berg put it, that biologists everywhere, regardless of their experience and specialization, he felt could create hybrid genes in a lab. The speakers at the conference often seemed to talk past one another, until a delegation of lawyers remind the attendees of the legal consequences that would ensue if any hazardous chemicals or organisms were to injure a lab worker or escape into the world. This reminder and fears of legal accountability convinced the organizing scientists that the conference could not end without producing some sort of regulatory statement. The final Asilomar conference statement, which you see here, was put to a vote and nearly unanimously accepted by the attendees. It included provisions for the regulation of different levels of biohazard, containment facilities for different types of experiments, and the requirement that all hybrid organisms be quote unquote crippled in such a way that they could not survive outside of a laboratory. The guidelines also recommended the continual revision of regulations as new information was discovered. The Asilomar conference was primarily concerned with whether rDNA technology could be safely researched and how experiments might be conducted. Whether such research ought to be done was a question the conference debated vigorously but left unresolved. The press coverage which followed in the wake of the 1975 Asilomar conference was unprecedented for a scientific gathering. Journalists who attended the conference helped extend the debate beyond scientific circles, enabling a larger segment of the population to participate in a public discourse about the scientific and medical promise of the new technologies, but also to consider the risks. Publications across the journalistic spectrum addressed the issues. In the pages of Vogue magazine, readers were asked to consider how the prospect of DNA research might change their lives, for better or worse. And in the New York Times, Stephen Tullman argued that the days of scientists deliberating in private over scientific issues of such consequence were over. He wrote, quote, "The need for improving the forums for negotiation between the scientific community and the larger public has been clear, at least since Hiroshima. The recombinant DNA problem is as much a symbolic as it is a practical issue," end quote. The 1975 Asilomar Conference on rDNA and DNA cloning was a watershed, a pivotal and transformative moment in the history of scientific practice, deliberation, and communication. It marked a major shift in how scientists, A, attempted as coordinated bodies to consciously monitor and regulate their own research and behavior, B, to consider the risks and ethical implications of their present and potential future research, and C, to debate these questions and share their conclusions in ways that actively engaged the broader public in discourse. To bring the impacts of this conference and its aftermath into sharper relief, it is illuminating to examine it against the historical backdrop of perhaps the second most impactful life sciences conference of the 20th century, one that gathered many of the world's leading biologists, social scientists, and politicians to consider the power of genetic manipulation to reshape humanity. On July 24th, 1912, the first International Eugenics Congress opened at the Hotel Cecil on the banks of the Thames in London. The hotel was an impressive setting. It was the largest hotel in Europe at the time and was often used to host important diplomatic or national events. The Congress was organized by the British Eugenics Education Society, and the attending elites represented the eugenic, race hygiene, and genetic societies of several nations, as well as members of governmental, administrative, religious, academic, medical, and legal institutions. There were even members of the military and representatives of feminist and social reform movements in attendance. The Lord Bishop of Oxford attended, as did Winston Churchill and Alexander Graham Bell. This was a high-profile event and a prestigious one as well. The eugenics movement had not yet acquired the negative associations it has today, and many of those in attendance considered it a progressive movement for social reform. The 1912 Congress in London was dedicated to Francis Galton, who coined the word eugenics in the late 19th century and conceived of the central ideas behind the movement. A half-cousin of Charles Darwin, Galton published a book in 1883, which built upon Darwin's theory of evolution and proposed an applied science which would employ human rationality to intervene in natural selection and improve the human species through the selective, quote-unquote, breeding of certain, quote-unquote, desirable individuals and the selective prevention of breeding by so-called undesirables. Galton's eugenics did not draw much attention, however, until a combination of social and scientific theory at the beginning of the 20th century helped to popularize his ideas. There was no widely accepted theory of heredity which provided a scientific framework to apply Galton's ideas until, around 1900, 3 different botanists independently rediscovered Gregor Mendel's previously overlooked theory of heredity. And this time, the scientific community took notice. English biologist William Bateson, shown here, was known as Mendel's bulldog. He was one of the most vociferous proponents of Mendel's theory around 1900. He proclaimed that Mendel's theory would grant humankind more power over nature than, quote, any other advance in natural knowledge that can be foreseen, end quote. Bateson was one of the first to recognize the potential that Mendelian heredity held for the eugenic movement, predicting the future with unsettling accuracy. Quote, when power is discovered, man always turns to it. The science of heredity will soon provide power on a stupendous scale, and that power will be applied to control the composition of nations, end quote. That time was not far distant at all, as we know. Changing social concerns at the turn of the 20th century in Europe and America, including middle and upper class fears of supposedly racially inferior immigrant populations, were coupled with the recently popularized theory of Mendelian heredity. This coincidence of social and scientific factors produced incredibly fertile soil for the growth of the eugenics movement. The 1912 Eugenics Congress featured multiple papers on heredity and proposals for using control over breeding to improve society. The consensus view was that undesirable social traits were a consequence of, quote, bad heredity, and the supposed immutability of that hereditary information meant that undesirable traits needed to be eliminated through controlled breeding, rather than through social policies that might have focused on factors such as education or social welfare. The very few dissenting voices at the Eugenics Congress in 1912, who argued that the impact of environment might be more important than germ plasms and hereditary traits in shaping society, these critics were drowned out in enthusiastic acclaim for the power of eugenics to make a better world, without posing any risks to human safety or health, it was argued. Major Leonard Darwin, Charles Darwin's son, concluded the Congress by stating to thunderous applause that, quote, through science, it is now within our power that civilization will be progressive, not retrograde, end quote. In the decades that followed the 1912 Congress, leading scientists and policymakers in Europe and the United States pursued eugenics policies in a quest to realize Major Darwin's vision. These efforts, of course, were eventually and horrifyingly expressed in the Nazis' pursuit of so-called race hygiene measures to allegedly purify German populations, to create a master race rid of the undesirable features carried and reproduced by so-called non-Aryan peoples, homosexuals, communists, and other undesirables. The Nazis' gruesome extension and application of eugenics theories that had once found scientific respectability reviled the Western public in the years after World War II. The growing recognition that such theories and practices, once thought to be scientifically sound and promising, could in fact lead to intolerable risks and frightening hazards, was a common feature of public scientific discourse by the mid-20th century. The idea that science, along with being a source of benefits to humankind, could also engender the frightening prospect of thermonuclear war, or could lead to the poisoning of the environment, began to shape the thinking of a post-war generation of scientists who, in the decades that followed, were increasingly inclined to train a critical lens on their own scientific fields and practices, even as they made astounding progress in their academic and industrial laboratories. It is in this light that we ought to consider the significance of those pivotal moments in the early to mid 1970s, when the same scientists who engendered a revolution in chemistry and biomedicine through the invention of rDNA technologies also paused their research efforts to gather at Asilomar. There, they deliberately, proactively considered the harms and the risks, as well as the staggering potential benefits that their work could create. Today, on the heels of last year's Third International Summit on Human Genome Editing, we would do well to remember that our current efforts to harness the potential of the gene editing revolution, while simultaneously managing its commercial, political, and health challenges, have their roots in these pivotal and transformative events 50 years ago. And it is with this thought in mind that the historians of the Science History Institute, my institution, in collaboration with colleagues at Stanford and Rice universities, will convene another conference at Asilomar in February 2025. On that same Monterey shore and in those same conference halls, our goal will be to revisit the heady debates of February 1975, drawing historical lessons that we hope to apply to the compelling life sciences questions of today and tomorrow. Thank you very much. And if there are questions,
David, I have one or two questions I want to ask you before I open it up to the audience. And we have another 10 minutes before the end of the 1 hour session, and then we'll go into the fluid space. So you've done a great job of explaining the history and the invention, but also the issues that scientists and the public and government will have to deal with regard to the boundaries around DNA research. I mean, DNA is not the only technology this faces. Let's remember that now AI, we're going through the same issue, which is what are the guardrails that are necessary for AI going forward, so you get the benefits but without some serious damage to society, et cetera. And so this is not a new thing. But what I'd like to do is turn from history to the future and ask you two questions regarding the future. One, what do you feel? I mean, DNA research continues to evolve, and there's more and more innovation that is applied to lots of different problems. So the first question is, as you look forward the next 5-10 years or so, what do you think are the new developments in DNA research that are going to become real, and where they're going to apply in terms of their benefit? But the second is, what do you predict will be the way in which these ethical and other considerations are going to get resolved and regulated, not only in each country, but also on a global basis, because it's a serious problem. You know, we know that there are all sorts of things happening. There's one scientist in China that was doing some experiments that caused a huge uproar. Now, he was discredited afterwards, but it created a spotlight on what the problem is. So the first is, what do you see the new innovations for DNA in the next 5 to 10 years and where they're going to apply? And the second is, where do you see the guardrails, and how do you think that ultimately this will get regulated if it gets regulated at all, because maybe your view is it's going to be hopeless to try to regulate it. So those two questions I'd like to pose.
Yeah, let me start with the second one first. It's interesting to note, first of all, I want to circle back to the example of He Jiankui, or better known as JK, the Chinese scientist who I referenced at the 2018 gene editing meeting in Hong Kong. After the disaster of his presentation at that conference, where he described using CRISPR to edit embryos and then produced actual births based on that technology, he was discredited. And the Chinese government was so appalled and embarrassed by this that he was actually tried and sent to prison in China for 3 years, beginning in 2019. Now, the interesting thing is that he has been released. He was released in 2022 after serving a sentence, and he is now rehabilitated and is working in another cutting edge biomedical institution where he is pursuing CRISPR research in somatic cells now, not germline cells, but in somatic cells to generate applications, biomedical applications that would be beneficial to human health. So he is back in the game. But one of the consequences of that episode was that it created a bit of a fissure between nations around the question of how this work should be regulated. The Chinese were initially incredibly proud of his work, then embarrassed by the hue and cry over it internationally, and then quick to embrace him again and that body of research, and to invest in it in China to create new technologies for human health, and also to create competitive economic advantage in the life sciences. So what does that say about where regulation is headed?
Right. Although that's, to be fair, David, that's only one example of many, many situations. So I'd rather not just fixate on that one. That's an example of a problem that occurred. But speak globally without fixating on that one example of what's happening in terms of overall the regulation of DNA research.
Well, this is what I'm saying. So there were hopes at that time, say the period 2015 to 2018, that by convening these meetings, that scientists and government officials could come to not just national consensuses, but an international consensus about what would be permitted in gene editing technologies and research. And in fact, that international consensus has eluded us. And there are approximately 70 countries now, so not all countries in the world, but 70 countries in the world that regulate this work, but they regulate it in different ways and at different levels. And in fact, the United States has a less strict regulatory regime than multiple other countries do, Canada and India, for example. And so I think the takeaway is that there is no strong consensus, and Jennifer Doudna is one who would say that there's no immediate prospect of achieving an international consensus about how to pursue gene editing research and applications going forward. Now, in
other words, there are no guardrails except maybe individual ones that each country might want to put in for their country, essentially. So in other words, it's chaos.
Well, it's controlled. I mean, it's controlled chaos. I think because the other guardrail you have is, I mean, first of all, there are some countries that are fairly heavily regulating this, but the other dimension is that what we have achieved, I think, is a fairly strong scientific consensus that can kind of feed self-regulation on the part of scientists, that this kind of work has to be closely monitored. No one is prepared to say yet that we should just sort of unleash every new application, for example, in germline editing. That is still, people are still treading cautiously there, and they're not prepared to say that in every dimension it's ethical. And so I think that, again, building on what happened at Asilomar in 1975 and the scientific culture that developed over the following 50 years, there is a strong scientific consensus, particularly in the strong research labs that matter, that we have to tread very carefully here. Now, you also asked about, where are the exciting areas where this technology, or where biologists, biomedical specialists are excited today. I mean, you mentioned AI. I certainly hear a lot of excitement around AI, particularly as a way of.
No. I want to talk about DNA research. In other words,
Right.
what are the new developments in DNA research that you see materializing this 5-10 years or so?
Yeah, I would
DNA technology.
I would say, I mean, I could say that certainly Jennifer Doudna and Feng Zhang and others at the Broad Institute, who are at the leading edge of this, really see a lot of potential in using CRISPR gene editing techniques in the microbiome. You know, we're all learning that there's a lot of potential in analyzing gut bacteria, and it's in the microbiome where they see the potential for tremendous therapeutic progress in years going forward. So that would be a key area. And the other thing that I think that is a form of or a source of excitement is that the cost of this is coming down dramatically. You can now essentially buy the materials you need, the CRISPR materials you need to conduct these experiments and interventions for a few $100s. So the cost has come down dramatically, while the efficiency and the precision of the targeting has gone way up. And so this is going to enable a lot of exciting work to take place, particularly around the microbiome. And I know that that's where the Innovative Genomics Institute, which is headed by Jennifer Doudna, is focusing a lot of its attention going forward. But again, guided very much by the history of these developments going back at least 50 years, and with those ethical concerns in place.
So we're a few minutes away from the 1 hour mark. I don't see any questions by the audience, so I'm going to pause and say, if anyone has a question before we head into this kind of virtual networking, I'd encourage you to type it in the chat box, and so that David can address it. Are there any questions?
I'll just say, while you're waiting for that, Pete, I'll just say that one other area of this that I really didn't have time to get into this presentation, but I would love to say more about it, perhaps casually, is that the history of how the intellectual property and commercial competition around gene editing developed is really fascinating. And much of one of the key features of what's happening in the CRISPR gene editing space today is this really ferocious competition between individual scientific labs and the ferocious pursuit of intellectual property, of patents, and the commercial advantages that those can secure for different scientific teams and the biotechnology companies that want to apply this technology for profit.
And in fact, there was a huge elite battle over the intellectual property rights related to CRISPR between Doudna and others at MIT, et cetera. But it took a long time to resolve, and I'm sure there'll be more that come up as there are more and more innovations around the whole DNA research.
Well, that's right. And in fact, it's still not resolved. And in fact, the Feng Zhang team at Broad Institute in Cambridge, Massachusetts, ended up, 8 years later, actually securing the principal patent, the first to file patent, so to speak, as opposed to first to invent, which has enabled them to block some of the commercial work that the Doudna group has wanted to undertake. You could argue that the Doudna group got their just desserts, though, because Doudna and Charpentier won the Nobel Prize. So how do you want to be?
We have one question. There's only one. So
Yep.
I'm going to read that for the audience, and then David, if you can answer it. And then once you've answered that, we're all going to go into the virtual networking event. So the question comes from Margaret Caldwell, and her question: has CRISPR been used in immune deficiency research and ways to get a better functioning immune system?
The answer is yes, but I don't really have the particulars on that. I know that what I can tell you is that of the historical characters that I've been talking about today, I know that group at the Broad Institute, led by Feng Zhang, has made significant progress in this area, and also I think a group of researchers at Cambridge, University of Cambridge, So perhaps if you wanted to communicate, Margaret, offline, we could talk a little bit about some of the leads that maybe could be followed in that respect.
So, by the way, though, David, thank you so much for a very insightful presentation. I think for those who aren't familiar with the history of DNA research and recombinant DNA, this has been very illuminating. I've got a couple notes here from people saying great presentation. So with that, I want to thank you, David, for that presentation. I'm now going to move everyone into the fluid space, and we'll have some fun talking to each other. You can say hello to David. And what I'll do is I will end this session. David, you're going to go into the backstage and then exit the backstage with the arrow at the top, brown arrow at the top.
Okay.
And then we'll all go into the fluid space. So we'll look forward to seeing all of you in a minute or so in the fluid space. And I'm sorry there are no cocktails, but there'll be lots of good conversation.
Very good.